The University of Wisconsin Zoological Museum was established in 1848 to be a center for research and educational support for the University of Wisconsin-Madison. It is dedicated to the preservation, study, and understanding of the vertebrate and aquatic fauna of Wisconsin, the Midwest, and other parts of the world. The museum acquires, possesses, and houses documented specimens resulting from field and laboratory research and the work of contributing agencies. Specimens are available for use in research, teaching, and outreach.
Hi, I'm Laura Monaghan, and I'm going to give you a very brief tour of the Zoological Museum, specifically the bird and mammal collections. This is our bird and mammal skin range. We're just going to talk a little bit about the mounts that are up on top of the cabinets, and of course we have inside the cabinets skins of mammals and birds. Here is a drawer of study skins with the associated skulls. Third area, again, the mounts up on top of their faces, and they have bags over them to protect them a little bit from the intake, the air intake. And here we have some larger birds. And then some smaller birds. And I'll show you these. We have, I think, 7 passenger pigeons. So in this museum, we have about 28,000 mammal specimens, about 24,000 bird specimens. In the skins, there are about 12,000 mammal skins and about 10,000 bird skins. The remainder of the mammals and birds are in the Archaeological Collection, which we will tour next. So this is the osteology lab. Most of our small bird and mammal skeletons are in this room. The birds are on this side of the room. You can see how things are organized in here. We have about 20,000 skeletons of all vertebrate classes. And we skin and deflesh them here, and we'll visit the prep room in just a couple of minutes. And so birds are on this side of the room, mammals are over here. I'll show you another box of mammals. This is a cat.
The specimens on the shelves are articulated specimens. They have been put back together after being cleaned and prepared. Articulated specimens are very useful for exhibits and for outreach. This box contains a disarticulated specimen. Disarticulated specimens are often used by researchers.
Hello, my name is Emily Halverson and I'm the registrar here at the Zoological Museum. Part of my job is to keep records for the museum, which includes accessioning new specimens, cataloging, and capturing data to input into our database. On average, we get about 1,000 new specimens each year, and we receive those specimens from researchers on campus, as well as organizations such as the U.S. Fish and Wildlife Service, the Wisconsin Department of Natural Resources, and surrounding zoos and wildlife centers. Another part of my job is to keep permits for the museum. So we hold local, state, federal, and international permits, and those allow us to collect and retain all the specimens that we have in our collections. Specimens you see here are passenger pigeons. Passenger pigeons are now extinct, but they were native to North America, including Wisconsin. These passenger pigeons were some of the last specimens that were collected from Wisconsin, and they were collected in the late 1800s. You can see there are differences between the feathers. These are adult passenger pigeons, and these are juvenile passenger pigeons. Passenger pigeons are notable because they were the first example we have where we know exactly when the species went extinct. The last passenger pigeon was housed at Cincinnati Zoo, and it died in 1914. Her name was Martha. These specimens are long-tailed weasels. They're a great example of why museums collect more than one individual of each species. Even though these are all the same species, you can see the differences because some were collected in the winter and have white coats, and some were collected in the summer and have brown coats. You can also see that each of the specimens has a specimen tag attached to them, and that records where the specimen was collected, when it was collected, and who collected it. Museums also continue to collect specimens so that scientists can learn more about the species and can track changes over time by examining the data that goes with them.
Welcome to the prep room, where we prepare skeletons that are destined for the collection. After all muscle and viscera are removed, the bones dry out under a hood that keeps air circulating around the bones to dry them out. After all muscle, viscera, and tendons are removed, the bones are separated and left to dry under a hood which keeps air circulating around the bones. This ensures that they dry out properly before they go to the beetles. Our dermisterium is in another building on campus, where the beetles are happy to sit and chew on skeletons to their heart's content. These beetles are dermestids, or hide beetles. The media will call them flesh-eating beetles, but that's not really what they are. They don't actually eat living tissue. They will only eat stuff that's really dried, like jerky. The species of beetle we use is Dermestes maculata. Museums across the country use these beetles for their skeletonizing work. It's really the larvae of these beetles that do the most work. They are very small. They can rasp away at the smallest pieces of dried tissue that might be left on the bones until they are clean enough to return to the museum. Once back at the museum, the bones are frozen to get rid of any remaining larvae, so that the bones can then be soaked in a soapy water solution, and then dried, and finally boxed up and ready for labeling. In our collection, every bone of a skeleton is labeled with its catalog number. That way, if bones are taken out for comparative study, they can go back into the correct box. Very tiny pens, called rapidographs, or microns, are used to label the bones with a permanent ink.
Hey everybody, my name is John Lyons, and I'm the curator of fishes here at the University of Wisconsin Zoological Museum. And today I'm going to give you a quick introduction to the fish collection here at the museum. And I'll talk a little bit about what the fish collection is, what it consists of, and more importantly, why it exists and what the purpose is of the fish collection. So let's take a look and start looking at the fishes that we have in the collection. Well, I'm here in the basement of Nolan Hall on the UW campus where the fish collection is located. And what the fish collection is a vast collection of jars of preserved fishes, fishes that have been caught over the last 120 years or so and preserved for posterity so that we can use them. And you can see that there are lots and lots of jars. In fact, in the entire fish collection, there are about 20,000 jars of fish. And you can see that many of these jars are full with lots of different individual fish. And we can do a rough estimate, and there are probably on the order of about 250,000 different individual fish preserved in the collection. Now that's a lot of jars and a lot of fish, and it sounds kind of impressive, but in the big scheme of things, compared to all of the different fish collections and natural history museums around the world and around the United States, I'd say the UW Museum is kind of medium in size. It's not tiny by any means, but there are many collections that are much bigger. And in fact, the really, really large collections like the US National Museum or the British Museum, they have on the order of 500,000 jars of fishes and maybe 10 million specimens, 5 million specimens. So they're gigantic. They occupy whole buildings. So let's explore the collection a little bit further. So here in the museum, we have lots of different kinds of fish. Some of them you may be quite familiar with. Some of them are rather obscure and not well known. So for example, here's a jar full of baby sharks. This is a type of shark called a dogfish shark, Squalus acanthus, found on both the Atlantic and the Pacific Oceans on the coast of North America. If we move over here, we see some Wisconsin species. So we see a species found in the local lakes here in Madison called the long-nosed gar, a very long, elongated species that's a predator on other fishes. You can see that here. We also see things like a bowfin, which is another predator. And we see all sorts of lesser known species, things like shad here, which are a small silvery fish that are very important as food for other fishes. And we see different types of trout and salmon. If those of you who might like to go fishing for trout, we have collections of different kinds of trout, different kinds of salmon. Now, again, we have thousands of jars of fish, and in many cases, we have dozens of jars of the same species. And so that begs the question of why do we have all these fish? Why do we need all these fish? And so let me talk to you a little bit then about what the purpose is of the fish collection and fish collections in general, and why we need and want to have all these different kinds of fish. So why do we have the fish collection? What is its purpose? Well, all of these preserved fish that are here in the shelves and in these jars serve a variety of roles. They are used for teaching here at the university, so we can teach people about the anatomy of fish, how to identify fish, the structure and function of their bodies, their different appearances. We have fish from all over the world. The majority of our collection is from Wisconsin and deals with Wisconsin species, but we have fish from coral reefs, we have fish from Africa, we have fish from the Amazon River Basin, we have fish from all over the world. And that gives us a good idea of the biodiversity of the world and allows us to compare and contrast what we have here in Wisconsin with other species in other parts of the world. And these preserved fish can tell us a lot of information. You know, they don't look very exciting. They're obviously dead. And in most cases, they've faded. They've lost their colors and vibrancy that they have when they're alive. But nonetheless, they are a treasure trove of information. So we can tell from these, of course, what species it is, what kind of shape it has. We can have information about what the fish is eating, how old it is, how fast it grows. We can use them to look at what sorts of toxic pollutants might have been in the water they inhabited, how they interacted with other species, all sorts of interesting scientific and conservation questions that we can use to preserve fish. The other thing that the museum is important for is it's a record. It's a record of our past. It tells us what was found in a particular area at a particular point in time. So each of these jars has a label, and it gives the species name, and then it gives the location where the fish was caught. It tells the date when the fish was caught, and it tells who collected it, among other things. And then it has a unique catalog number so we can identify it and track it. And then with that number, that's associated with other files we have that provide other information about how and where and when and why it was collected. All that sort of information makes the collection kind of like a giant library. Scientists and students and the general public, for that matter, can come in and see what was found where. It's a record. They can actually check out specimens to look at in more detail under a microscope. And so it's a record. It's an archive of what the biodiversity of our waters and waters around the world have been over time. And in many cases, we can show that, long ago, there were different species found in particular areas. And that in many cases, we've lost certain species. They've been eliminated. In some cases, have gone extinct. In other cases, new species have arrived and invaded and changed the ecosystem. So this provides a permanent record, a way to confirm, in fact, what was actually there. There can be no disputing because we'll have an actual specimen. You can say, yes, indeed, even though a species is not here now, it once was here. And that tells us something about what the condition of our waters is now and how they've changed over time. So I hope you've enjoyed this tour of the Zoological Museum and the fish collection specifically. It's an active collection. We're working and trying to make it bigger and better every day and make it more accessible, the information that it contains, accessible to scientists, conservation, and students like you and the public. So please consider visiting the museum when things are a little bit better in terms of COVID, and we look forward to seeing you. Bye-bye.
Hi, welcome. My name is Jason Mechanic. I am a museum scientist here at the UW Zoological Museum. And my work here at the museum is primarily to catalog and identify bones from archaeological sites in Alaska. Wait, why does a museum in Wisconsin have archaeological bones from Alaska. Well, come with me and we'll find out why. You see all those boxes behind me? Those are from archaeological excavations that happened in the 1950s and 1960s by William Laughlin and William Rader in the Departments of Anthropology and Zoology. These excavations took place in the Aleutian Islands. You may or may not know this, but museums often act as a repository for archaeological material, meaning that when people go and excavate sites, a lot of the materials from those excavations end up here in museums. Some of these excavations may have happened last year or 2 years ago, but often these are from excavations that happened several years ago, in this case, in the 50s and 60s. These are legacy collections. So legacy collections are old collections that were previously excavated. So fossil remains are animal bones. So people who were living thousands of years ago were collecting and harvesting animals. And if you come with me to the lab, we'll kind of talk about what these actual animal bones can tell us about past lifeways and current climate change and why it's so important for us to catalog and identify these species. The Chiluga site is an exceptional archaeological site in the Aleutian Islands on Umanak Island, the ancestral home of the Aleut people. The site dates back to around 4,000 years. The site itself is a very deep midden deposit, which is archaeology speak for garbage pile. These refuse pits, these garbage piles, are extremely valuable to archaeologists because people will leave behind things they hunted. And the bones that people leave behind will give us an idea of how they were making a living. So what kind of animals were they eating? The environments that they were living in and the season. But also gives us an idea of how climates change and species changed through a 4,000 year time period. So people at the Chiluga site ate a very diverse diet of different foods. Here from this layer, we see a really kind of rich, diverse snapshot of some of the foods that people were eating. And this is only a small little sample. So we see limpets, which are intertidal invertebrates that people were collecting on the shorelines. We also see dogwinkle as well. And we also have species that are quite bigger, such as sea lion and seal. And we also have a lot of fish remains, such as the Pacific cod right here. Apart from learning about past environments and lifeways, zoological and archaeological material is really important because it can forecast and demonstrate how species were changing in response to prolonged hunting pressures or due to climate change. One research question of interest is looking at how fishing and climate affected Pacific cod size after 4,000 years of human occupation. Here you're looking at the dentary. The dentary is a mandible, a fish mandible. Here we see the teeth, there, and we see that they come in different sizes. So these bones can be used to reconstruct past catch data, as fish length is highly correlated to bone size. So looking over a 4,000 year period, we can see how fish length decreased through time. This data is extremely valuable for modern fisheries, as it provides a baseline on past fish lengths, but also gives an indicator of how fish species, such as the Pacific cod, may respond to warming and cooling periods, and will hopefully help in current conservation efforts. So, as we can see, there's a lot that these old museum collections can tell us about past populations, but also hopefully help us in learning about modern climate change and kind of helping modern species. But before we can do any of this, what we need to do is identify and catalog these legacy collections into a database so the data is organized and can be accessible to future researchers who can then study the material. Thank you guys very much.
A little known part of the Zoological Museum collections are the Historical Instruments collection. This collection houses a range of old historic instruments, from scales, antique microscopes, beautiful wax models, and historical wall charts that were used in teaching for many years. The museum even has a few Blaschka models, which were used in teaching in the early 1900s. These are delicate models of aquatic invertebrates like jellyfish that were made in the 1800s by a pair of European artisans. These models are considered the peak of glass artistry, and their techniques are very difficult to replicate.